The virus was originally isolated from lung wash from patients with pneumonia of unknown etiology in Wuhan, China. They used tissue culture to amplify the virus and allow for electron microscopy. They also used standard multiplex PCR type assays. These are molecular probes that recognize all conventional viruses. And these days we also have the chance to use deep sequencing to look at the entire material and identify anything unexpected or novel in that sample. And here we can see pictures of the electron microscopy results showing a virion with those little spikes around the outside. And those are typical of coronaviruses. The multiplex PCR interestingly showed similar results identifying a coronavirus but couldn't identify a specific type of coronavirus, so it was novel. And our deep sequencing identified something that was unexpected in humans and not seen before. The sequencing technology is rather complicated and difficult, and moving it from that clinical sample into a nice conventional genome that we can all look at is not easy. But in China they did that very rapidly, and luckily they published that online so that we could all see it. And this is a little process of that sequencing technology going from the original genome, which is fragmented small pieces of sequence, the short reads, and then recompiled into this genome. And this is a 30,000 base pair genome that you can see, which is pretty large for viruses. And if you look along all those gene names, you might recognize the spike protein, which is the receptor binding and how the virus enters human cells. When we have that genome, we can now start to put it together with all the other viruses. And here you can see all the coronaviruses that we know about put together. And you can see SARS-CoV-2 there, along with the bat viruses, not too far away from the SARS coronavirus. We can see that zoonotic viruses that occurred since 2003 is the MERS coronavirus, SARS coronavirus, and now the SARS coronavirus 2, which is the cause of COVID-19. We can also see in this tree the so-called human coronaviruses, which are commonly circulating in humans, and those probably originated from rodents, some in the historical past. If we look more carefully at the viruses most similar to SARS-CoV-2, we can see that they originated in horseshoe bats. And there's quite a diversity of these coronaviruses in horseshoe bats, and SARS-CoV-2 fits right into that. However, there's a little bit of evidence that some of the SARS-CoV-2 virus looks like a pangolin coronavirus sequence. And that suggests that potentially the virus passed from the horseshoe bats into the pangolin and then from there into humans. But now the virus is really in humans, and there's a diversity starting to build up. From the more than 3 million infections that we know about, and there's probably more than that, we've managed to whole genome sequence more than 5,000 of those. And here we have the tree of 5,000 genomes. You can see that there's a little bit of clustering of those. We've clustered the cluster 1, the original, in green here, and cluster 2 is in the reds, orange, and browns. But these genomes are not very far apart from each other yet. That scale is one single nucleotide polymorphism, so it's only one SNP apart, and most of these genomes are just a few SNPs apart. However, this genetic diversity is extremely important. We are expecting more diversity to generate over time. But even right now, those diagnostic primers that we designed to that very first genome may not match all of these 5,000 genomes that we have, or the diversity of genomes which are out there already. And over time, these might even build up potentially resistance or divergence in their response to therapy and even to the vaccine. In this picture, we have a Bayesian time-dated tree, and you can see back in January, the number of viruses was relatively small, but by March, this tree has 1,200 genomes, and you can see that diversity increasing rapidly. And we've colored it by the different continents, and you can see there's some strains are more common, for example, in North America, and some in Europe by the different colorings. And this might mean that the diagnostics, for example, might need to be a little bit different depending on where you are. luckily, there's a worldwide effort to keep track of all of this diversity, and we look forward to seeing how that generates in the future, and we can all keep an eye on it. I'm very confident that we won't see too much diversity, as this virus seems very well adapted to humans, but we need to keep an eye on that.